Afferent arteriole comes in and it pours into the glomerulus. This entire structure of capillaries, fenestrated capillaries, is the glomerulus. And the glomerulus is covered by these cells. Now this model makes it look like it's only half of it's covered in cells, but really the whole thing is covered. They just don't show them on this side so you can see the detail of the simple squamous that's forming the glomerulus. These cells that are covering it, this is referred to as the visceral layer of the glomerular capsule, also called the Bowman's capsule.
So it's... Oh, hang on. So the Bowman's capsule is there and not here?
This whole thing is the Bowman's capsule. This is the parietal layer and this is the visceral layer.
Okay.
So this is the visceral layer of the Bowman's capsule. They serve the same function as astrocytes in neural tissue. They create a blood barrier that doesn't let everything drain out of the blood. Only certain things, mostly toxins and stuff. So that's the whole point here for the glomerular capsule, is to get the blood out of the capillary and into a unit of the kidney where we can actually process the blood. So it's all draining out of the capillaries into the Bowman's capsule. And this keeps certain things in the blood that we don't want to lose, like red blood cells. Things like that. Little important things.
Those little blue things on there? Or
no, that
the whole
blue thing is just the nucleus of the cell.
Oh, okay.
The whole thing. These red lines are the borders between one cell and another. So this yellow tissue, this whole thing is a cell. And these cells compose the visceral layer of the glomerular capsule. This is the parietal layer of the glomerular capsule. And it's collecting all of this filtrate that's coming out of the blood, and it passes through these brush border cells. And that's one of the first levels of filtration, basically. So we squeeze out all the blood into the capsule, or all the fluid from the blood into the capsule, and we're passing through these brush border cells, which have a lot to do with how we're filtering, how we're absorbing, reabsorbing, secreting, etc. You'll go more into detail about that in your physiology class, about what is traveling in and out at what point. But this is the beginning of the proximal convoluted tubule. You can see the proximal convoluted tubules here. If we look at one of these open glomerular capsules, you can see where the afferent arteriole comes in. Here's the glomerulus, here's the Bowman's capsule or the glomerular capsule, and here's the proximal convoluted tubule. After it passes through the PCT, it'll drop into the loop of Henle. The loop of Henle has a... Yeah, go ahead.
How can you, just by looking at this, how do you know that's the afferent?
Because this is coming off of the cortical radiate. And afterwards, you see it branches out into the tissue of the cortex itself. Right. So after it passes through the PCT, it'll drop into the loop of Henle. The loop of Henle has descending limbs and ascending limbs, and it has thick limbs and thin limbs. So like this one shows it really well, where you have a thick descending, thin ascending, thick ascending. After it goes through the loop of Henle, it'll come up and it goes into the distal convoluted tubule. And this is going to pass right by the glomerular capsule, always. Because our first level of filtration is here, where we secrete the blood, or the fluid from the blood, into the glomerular capsule. Our second level of secretion, or filtration, our second level of filtration is the loop of Henle itself, where one side you have semi-permeable membranes around it, where you have on one side fluids coming in and out, and you have solutes coming in and out on the other side. And again, you'll study more about that in physiology. Right now, you don't have to know the specifics of it, but just know that we're straining the fluid and removing what we want to keep, and keeping in the fluid what we want to get rid of. After that, the filtrate continues coming up and it passes by the Bowman's capsule again. Here it's passing through this triangle here between the afferent, the DCT, and the efferent arterioles. All of this together forms the juxtaglomerular apparatus. Breaking down the words, juxta means next to, juxtaglomerular apparatus. It's the apparatus next to the glomerulus. Some people make the mistake of thinking the whole thing is the juxtaglomerular apparatus, and it's not. It's just these three and the tissue in between that are dealing with that. And this is the final level of filtration. We're going to filter the fluid between these one last time. And then what's left in the filtrate, that's what we're going to get rid of. That pours from the DCT into these collecting tubes. At this point, it's urea. Or no, I'm sorry, it's not urea yet. It's urea when it exits the collecting tube and enters the calyxes.
It's urine.
Yes, it's urea here. It's urine when it passes there. Okay, so it's urea here, and we've finished filtering it. It's now urea. We pour the urea down, and it goes through the collecting duct into the minor calyx. At that point, it's now urine. So the urine is collecting in the minor calyxes. The minor calyxes have main branches that they come together to form. Those main branches are called the major calyxes. The major calyxes all come together in one large chamber. That is called the renal pelvis. So from the renal pelvis, it exits the kidney into the ureter. And the point where the renal pelvis becomes the ureter is when it passes the hilum. Now this is a portal. A portal for an organ is basically blood vessels and everything going in and out. It's not the same as a portal system that you'll see in capillaries, but it is a portal, and it's identified by the hilum, which is the opening of the organ itself. So this opening is called the hilum, and this is where your blood vessels are coming in, blood vessels are coming out, and your ureter is coming out. So at that point, it transitions from renal pelvis to ureter. Your ureter is going to drain into your bladder around the trigone area, and that's going to go through the urethra and be expelled from the body. That is the path of a filtrate moving through here. Now, you have two types of nephrons, because this entire structure is a nephron. You have nephrons that just come a little ways into the medulla, and you have ones that come all the way into the medulla. And that's going to be your cortical nephrons, because it's primarily in the cortex, and you're going to have your juxtamedullary nephrons. And those ones, again breaking it down, juxtamedullary, next to the medulla. The glomerulus and the nephron as a whole is next to the medulla. And the reason why we have this is because we can drop farther, and as you go down through the pyramid, as you go down into the medulla, you're going to reach higher solute levels of the interstitial fluid, so you're going to reach a higher saline content. And that's what powers the filtration through the loops of Henle. So this is not nearly as common. These are far more common. But we do have both.
Does the blood pressure play a role in that motivation to there?
A little, but not a lot.
So what is it? What is
The primary motive force is the saline content. And as you drop down, the saline content increases pretty much exponentially. It's extremely salty down here. And it's a little salty, but closer to normal body levels up here. Okay. So you have your cortical nephrons and your juxtamedullary nephrons. So called because this is the glomerulus itself, and the main part of the nephron is closer to the medulla. Now, this is the cortex of the kidney, this is the medulla. So let's look at this larger thing to see the overall gross anatomy of the kidney. Gross anatomy, if you will, at least. So you have the cortex all the way around. This is where the nephrons, the main body of the nephrons, is located in the cortex. When you pass into this level, you're going to have the medulla, and this is formed by your columns and your pyramids. Your renal pyramids are going to have your papilla towards the base, and they're going to have the loops of Henle through the middle of them, and the collecting ducts right through the center. Other than that, you have to know the blood vessels coming in. This is the renal artery, and the renal artery is going to split off, and it's going to go into your interlobar arteries, because they're going in between all of your pyramids. As they come up, they're going to pass across the top of the pyramids, just under the cortex, and those are going to be your arcuate arteries as they arc across. Then they're going to branch off and shoot up through the middle of the cortex, and those are going to be your cortical radiate arteries. It's from the cortical radiate arteries that we get our afferent arterioles coming off. They'll go into a capsule, and then they'll come out as efferent arterioles. The efferent arterioles will eventually turn into your venous system and drain it. So here you can see your arcuate artery, you can see your interlobar artery, and you can see your cortical radiate arteries. And the outside edge, that's the renal capsule. I think I hit everything.
I think so.
Thank you.
It is a lot of